Hello everyone, welcome to ITLS Academy. I am your mentor Dr. Shamaita Basu. So in this session we will be talking about part 2 of the topic genetic basis of disease. So I completed with the uh, chromosomal abnormalities. I think we last talked about in the last session cystic fibrosis and Huntington's chorea. So here in this second session I would like to start with single gene disorders. Okay. Now, single gene disorders means those diseases or those disorders which are involving mutation of a single gene. So, under single gene disorders, we have various categories of defects or disorders. What are they? Firstly, what we have is enzyme defect, that is, which is also known as inborn errors of metabolism, under which we will talk about phenylketonuria, we will talk about alkaptonuria, and various other diseases. Okay. So, enzyme defect means there is a particular defect in an enzyme. And what is an enzyme? Enzymes are nothing but protein in nature which are acting as biological catalysts, means they are helping in speeding up the different biochemical reactions that occur inside our body with respect to metabolism. Okay, metabolism means what? Metabolism of carbohydrate, metabolism of protein, metabolism of fatty acid or lipid. Okay, so for that enzymes are very important integral part. Okay, and if there is some amount or some kind of mutation in a particular gene which is responsible to code for a particular enzyme, then what will happen? That particular enzyme will not be formed. Okay, or maybe it will be formed in a defective manner, as a result of which it will not be able to exert its function, it will not be able to function properly. Okay, so that is what is enzyme defect is. And if, say for example, the enzyme which is responsible for metabolizing carbohydrate in your body, if that enzyme is not produced, then what will happen? Obviously that particular carbohydrate will not be metabolized inside your body. As a result, you will become intolerant to that particular carbohydrate. Okay, so these are also known as inborn errors of metabolism, because obviously there is some metabolic error that is going on inside your body due to that particular enzyme defect, and moreover it's genetic, so obviously it's inborn. Okay, so from there we get the name of inborn errors of metabolism. So what will be the effects or what will be the outcome of these enzyme defects, no matter what enzyme it is? Okay, so firstly, there will be accumulation of substrate. Okay, because again the same thing, what is the function of the enzyme? To speed up the biochemical reaction. Speed up means what? It is acting as a catalyst. It is reducing the activation energy which is required to initiate that particular reaction. Now, what do you mean by a reaction? What do you mean by a, sorry, by a chemical reaction in the body? Means the enzyme will be acting on the substrate, on some reactants which are nothing but products, chemical products. Okay, it can be carbohydrate in nature, it can be lipid in nature, it can be protein in nature. So those reactants will be the substrate on which the enzyme will act. As a result, we will get the products. Okay, so the substrate will be converted to the product. Now, if that particular enzyme is not present, then what will happen? The substrate will not be converted into the product. So gradually over time, that substrate will be getting accumulated inside the body. Okay, so if that substrate is accumulated inside the body, then not only that particular substrate will not be metabolized inside your body, but also accumulation of that substrate in a huge amount will lead to toxicity, isn't it? Because any particular product which is getting stored and stored inside the body is not good for your body. It either needs to be used up or it needs to be excreted from the body. Okay, so that will be one of the major side effects of any enzyme defect. Secondly, there will be lack of product, because obviously the enzyme is not there, the substrate is accumulating, so obviously there is no question of product being formed, isn't it? So in that case also there will be a lack of product. Thirdly, there will be failure to inactivate a protein which causes damage. Again, very important. Say, for example, the function of the enzyme is to inactivate a protein which maybe is causing a damage or causing a disease. Okay. So in that case also, it will be a failure because the enzyme is not there. So there is no one to inactivate that protein, because maybe that protein, if the enzyme was there, then maybe that enzyme would be able to convert that protein into some other product or into some other uh, you know what to say, substance or compound, in that as a result of which the damage or the toxic effect that the protein was producing could have been surpassed. Okay. Second, what we have is, second we have receptor or transport protein defect. Example will be familial hypercholesterolemia. Now what is hypercholesterolemia? Hyper means increased. What is cholesterol? We all know. And amia, any particular term which is ending with the word amia, it means increased level in the blood, or it means in the blood, accumulation in the blood. So hyper means increased. So increased accumulation of cholesterol in the blood is what is meant by hypercholesterolemia. And familial means something which is passing on throughout the family. Means your mother has it, then you have it, then your children will have it, then your grandchildren will have it, like that. Okay. So that means familial, something which is running in the family. Means obviously something which is genetic. Then we have structural protein defect. Okay, structural protein defect may we have Marfan defect, we have Eldon defect. These are all named by the name of the scientist or the famous persons who suffered from this disease once upon a time. So the structural protein defect will lead to problems in the structure, problems in the function, and problems in the quantity of that particular protein. Okay. And enzyme defect, which increases drug susceptibility, is a very important example, or very common example is given over here. That is G6PD. The full form of G6PD enzyme is glucose 6 phosphate dehydrogenase, which is a very important enzyme as far as carbohydrate metabolism is like concerned. Particularly, it's the enzyme which is responsible for the process of glycolysis and also glycogenolysis. Okay. And the drug which actually is responsible to increase like the drug to whose susceptibility is increased due to this D6PD deficiency is primaquine. Okay, and primaquine is a drug which is mostly used in case of malaria treatment. Okay, so those patients who are suffering from this D6PD deficiency, those patients will be more susceptible to this particular drug as far as malaria treatment or any concerned treatment of this drug is concerned. Okay. Now coming to structural protein defects. Now what do you mean by structural protein? Whenever we are talking about protein, protein has three types of, or three stages as far as the structure is concerned. Three stages of structure. What are they? Primary, secondary, tertiary, and quaternary. Okay. So in order for a protein to be structurally active or biologically active, rather, means to be able to exert its function which it is supposed to do, it's very important that the structure of protein is properly formed. Okay. And moreover, quaternary structure of proteins will include examples like hemoglobin, myoglobin. Okay. So if these proteins, they do not achieve the last stage of the structure, that is if they do not achieve the quaternary structure stage, then in that case these proteins will be biologically inactive. Okay. And which factor will determine that whether these proteins will reach the quaternary structure or not? The enzymes, the genes. Obviously, if there is any kind of mutation in the gene, any kind of problem with the gene, then that will lead to structural protein defects. Okay. So the first syndrome or the first defect is Marfan syndrome. So here what is happening? There is fibrillin 1 defect, which is again a structural protein, mainly responsible for what to say, uh, structure of the cells, cellular structure. Okay. Uh, so it will lead to aortic arch aneurysms will be there, tall dislocated eye lens will be there. Okay. And some of the famous persons who suffered from this Marfan syndrome will include Abraham Lincoln and Osama bin Laden. Okay. And second structural protein defect that we have is Ehlers-Danlos syndrome. Okay, which is also known as AD or AR. So there are different types, around 6 different types of this particular syndrome, and they can be classical, they can be vascular. Okay. Dermatological, different types of types are given over here, and it will include various collagen defects also, like symptoms. and also it will be producing hyperelastic skin and hyperextensible joints. Means the joints will be extensively movable, which is again not a proper or a normal indication. Okay. Moving forward to receptor protein defects. Again, something very important. What do you mean by receptor protein? Whenever any enzyme or any hormone function is going on, or any particular chemical messenger is involved in any particular cell signaling or any particular cell function, it's very important that the proper receptors are recognized, because otherwise what will happen, the specificity will not be there. Okay, say for example insulin, it's a hormone. So insulin has a particular target site. Each and every hormone has a particular target site or target organ, isn't it? Now, how will that hormone or protein be able to recognize whether it has reached its target site or target organ or not? How? By recognizing the receptor. So if, say for example, liver is the site, the target site of insulin, then particular insulin receptors will be there on the surface of the liver. So the insulin will go and interact with these receptors. It will go and bind with these receptors, and thus it will send a signal that okay, I have come, now let me do my function. Okay, so this receptor protein is also a very important concept, and it is also a very important component of the normal physiological functioning of the human body. Okay, so firstly we have familial hypercholesterolemia among the receptor protein defects. So here what will happen? LDL receptor will be defective. What do you mean by LDL? Low density lipoprotein, which is actually a bad cholesterol for our body. Okay, it is a type of cholesterol which is bad. Like if taken in more amount, then it gets deposited in the arteries because its density is more. So it gets deposited. I mean, its solubility is less, so it will be deposited easily in the arteries of the heart, and it will lead to various cardiac defects, including heart attack, chances of heart attack. Okay, so LDL receptor में अगर there is any kind of defect, then it will lead to familial hypercholesterolemia. So the cholesterol transport across the liver cell will be impaired, and as a result, there will be cholesterol buildup in the blood. Okay, and there are some scavenger system also, which are known as the macrophages. They might help in uh, you know what to say, reducing or taking up or scavenging the cholesterol that is deposited inside the body. So the macrophages are known as scavenger system. Scavenger means taking up something. Okay, and that is also the reason why the macrophages are known as foamy, because while scavenging the the cholesterol, there's a coating, because cholesterol is nothing but it's a type of a lipid, so it will form a foamy layer. So since the macrophages are acting as the scavenger system, they will go and form a foamy layer on the macrophages. Hence the macrophages are also known as foamy. Okay. Now coming to enzyme defects. So by far the largest known category of genetic defects will be enzyme deficiencies, which will lead to substrate buildup, product lack, and the substrate buildup can also be harmful or toxic, as I already mentioned. Okay. And among the enzyme deficiencies, the most common will be lysosomal storage diseases, which comprise most of the enzyme deficiencies. Okay, so starting with the lysosomal storage diseases. So firstly we have glycogen storage disease. Means by the name only we can understand that there is some problem with the storage of glycogen in the body. What is glycogen? It is the storage form of carbohydrate inside our body, which is mainly stored in the liver or in the skeletal muscles. Okay, then we have sphingolipidosis or the gangliosidosis. Then we have sulfuridosis. We have mucopolysaccharidosis. We have mucolipidosis and various others, which also includes fucosidosis, mannosidosis, asperglycosaminuria, okay, and acid phosphatase deficiency. Now talking about glycogen storage disease, uh, there are many types. About 10 different types are there. Okay, among which prominent will be type 2, which is also known as pomp. Then we have von Gierk, we have McArdle, These are actually among the 10 different types. These three are actually the most studied and the most discussed. Okay, and the storage side, as I already mentioned, liver, skeletal muscle, and also heart. To some extent, in the heart also, it's stored. Okay, and this is actually a nice microscopic view of the different liver, muscle, and heart tissues where we can actually locate glycogen. So, this is basically the diagram of a histologically stained tissue. Okay, and whenever we are talking about histologically stained tissue, the two important stains that are used for any routine type of histological staining are hematoxylin and eosin. Okay, so toxylin will give you the pink color and eosin will give you the blue color. So as you can see over here, see small blue granules appear. So those are the glycogen storage deposits that you can see. Okay, and this pink color are nothing but the deposits. Sorry, the cells of the organ. Okay, which can be liver or heart. Secondly, we have sphingolipidosis. Okay, what is sphingolipid? It is nothing but a type of lipid. So if there is any kind of uh, defect or disorder with the storage of the sphingolipid, then it will be known as sphingolipidosis. Okay, so again we have different types, among which we have Tay-Sachs disease. We will be talking about Tay-Sachs disease, and it is most often referred to as Tay-Sachs disease is most often referred to as a type of sphingolipidosis. Okay, so here what is happening, gangliosides are getting accumulated. So see, again I have given over here a nice histological stain microscopic view. So again the blue deposits are actually basically the gangliosides which are getting deposited or accumulated. Okay, and why it is happening? Obviously we are talking about enzyme deficiencies, so obviously it will occur due to deficiency of some enzyme, isn't it? And what is that enzyme? That enzyme is hexosaminidase A deficiency. So the name of the enzyme is hexosaminidase A. Okay, so if this particular enzyme is deficient, then it will lead to this particular Tay-Sachs disease or sphingolipidosis. Okay, and it is particularly seen in Ashkenazi Jews. Okay, they are about 1/30th of them are the carriers. Carriers means they are just harboring the disease, but they are not showcasing any type of symptom. Means we cannot say that they are actually suffering from the disease because they are asymptomatic carriers. Okay, so they are just harboring the disease. Maybe they are just having one single copy which is defective of that particular gene. Okay, so they are not so showcasing any particular symptom, but mostly these Ashkenazi Jews are the sufferers of this disease. Okay, and CNS neurons are also a site of accumulation. Means if you go and look into the neurons of the CNS, means the central nervous system, which will include the brain and the spinal cord, then you will see that these sphingolipidosis or gangliosides are getting accumulated. Okay, and how will you be able to understand that? You will be able to understand that by a cherry red spot in the macula. So see, again I have given the diagram over here. So this is the cherry red spot that you can see in the macula. And what is macula? Macula is basically a part of the eye or where the rays are focused when we see. Okay, the light rays are focused on the macula. So this cherry red spot will be seen in the macula. Okay, that is a typical characteristic feature of Tay-Sachs disease. Moving forward, Niemann-Pick's disease or Niemann-Pick disease. Okay, different pronunciations are there of these diseases, because these are not scientific names. These are names behind the name of some scientists or discoverers or sufferers. Okay, so there can be different pronunciations. Okay, so Niemann-Pick's disease again have different types A, B, and C. And over here there is buildup or accumulation of sphingomyelin, which is a type of a muscle protein. Okay, so it leads to massive splenomegaly. Splenomegaly means enlargement of the spleen. Megaly means enlargement, and spleen refers to and spleno refers to spleen. Okay, and this is also very much common in the Ashkenazi Jews, just like the previous diseases, and it is often fatal in early life. Means death can occur in early life only, and there will be CNS accumulation of the sphingomyelin in the CNS, and also it will lead to organomegaly. Organomegaly means generalized inflammation or generalized enlargement of the organs. Not only spleen, but also other organs can also be enlarged. Okay, and again over here I have given the microscopic view of the histopathological staining. Moving forward to Gaucher's disease. Okay, so again it can be pronounced as Gaucher's or Gaucher's disease. So over here, the substance or the compound that will be accumulated is glucocerebroside. Okay, so glucocerebroside, by the name only we can understand that it is a type of a compound. Gluco means obviously carbohydrate type of compound which will be accumulated in the cerebrum. Okay, so right in the brain. Why? Due to the enzyme responsible, the deficiency of the enzyme responsible will be glucocerebrosidase. Okay, so glucocerebrosidase enzyme will be getting enzyme will be deficient, as a result of which its substrate, that is glucocerebroside, will be building up or accumulating inside the body. Okay, 99% are type 1, means again there are different types just like Niemann-Pick's disease, and there will be no CNS involvement. So this is a unique feature because in the previous two diseases what we saw was there is CNS involvement, there is CNS accumulation, the neurons of the CNS would have been affected, but in this case in Gaucher's disease there will be no CNS involvement. Okay, and all the macrophages, liver, spleen, nodes and bone marrow will be having deposits of this glucocerebroside. So again this is the microscopical view, and this is how typically your cerebrum, like the person who is suffering from this disease, his cerebrum will look like with the deposits of glucocerebroside. Okay, and then we have mucopolysaccharidosis. Okay, what is that? Mucopolysaccharidosis. Again, different types are there. So we have type 1 and type 2. They are respectively known as Hurler and Hunter. Okay. So the compound that will be accumulated will be darmatan sulfate and heparin sulfate. So these are the two compounds that will be accumulated, and the typical features will be coarse facial features, clouding of the cornea of the eye will be clouded, means there will be a hazy appearance, you will not be able to see everything clearly. There will be joint stiffness, so unlike excessive flexibility of joint which we saw in one of the previous diseases, in contrast in case of mucopolysaccharidosis there will be stiffness of the joints, means the joints will not be properly movable. There will be mental retardation also, and urinary excretion of these sulfates that are accumulated, that is heparin sulfate and darmatan sulfate, will also be one of the important clinical symptoms or features. Okay, and this is actually the typical appearance of how the persons who suffer from these diseases will look like. So see, from the face only we can make out that there are some coarse facial features, isn't it? Which is coarse means something which is abnormal or something which is too much exaggerated. Okay, so see the cheeks are broad coming up, the eyes are very much inside. Okay, the nose is not properly formed, the eyebrows are not properly formed. So these are the coarse facial features. Okay, and these are some of the other lysosomal storage disorders that we have. So these are actually not that much common and not that much important, hence I have not included them in details. So fucosidosis, we have mannosidosis, means accumulation of mannose. Fucosidosis is accumulation of fructose. Okay, aspartic glycosaminuria means accumulation of aspartic acid. Okay, acid phosphatase deficiency will lead to accumulation of phosphate esters. Okay, because obviously phosphatase means its substrate will be phosphate, so those will not be converted, so hence they will be, the phosphate esters will be accumulated. Okay, next, a very important inborn errors of metabolism that we have is alkaptonuria. Okay, so it is basically not a lysosomal enzyme disease, and it is first such disease to be described where homogentisic acid will be accumulated due to deficiency of the enzyme homogentisic acid oxidase. So the buildup of the substrate will be homogentisic acid, and the enzyme deficiency will be that of homogentisic acid oxidase. Okay, and the severe symptoms, the typical clinical symptoms will be black colored urine, black colored nails, which is also known as ochronosis. I will be showing you the diagrams. Black colored skin and black colored joint cartilage, which will lead to severe arthritis. Okay, why black colored? Because in all these places this homogentisic acid will start depositing or accumulating. Okay, So see, this is the typical appearance. This is just the microscopical view of the histopathological stain. See the ears, they are turning black or grayish in color. The nails are all turning blackish or grayish in color, and this is particularly the color of the urine, okay, in contrast to the normal urine. So this is, the left hand side is basically the normal urine of a normal person who is not suffering from this disease. So it's straw colored or pale yellow colored, as we all know, and this is basically the black urine of the patient who is suffering from alkaptonuria. Okay, again phenylketonuria, which I actually skipped in the previous session because I wanted to talk about it in the second session, since this is the session where we are talking about the inborn errors of metabolism. Okay, so in phenylketonuria, again, the product which is getting deposited is phenylalanine. Okay, so that is the substrate which is getting accumulated. Why? Due to the deficiency of the enzyme that is phenylalanine hydroxylase. Okay, so this enzyme is necessary to metabolize the amino acid phenylalanine to the amino acid tyrosine. So if this enzyme is not there, if this enzyme is deficient, then obviously phenylalanine will not be able to be converted into tyrosine, and hence this phenylalanine will go on getting deposited inside the body. Okay, as a result, what will happen? The symptoms will be mental retardation, head will be smaller in size and compared to normal persons, and also it can lead to epilepsy. What is epilepsy? Epilepsy is a neurological condition which leads to consistent and random seizures or fits. Okay, so these are the particular clinical features of phenylketonuria. Okay, moving forward to the last disease that I want to talk about in this topic of genetic basis of disease, that is neurofibromatosis. Okay, so neurofibromatosis, by the name only we can understand that it has something to do with the neurons. Okay, so again it can be of different types, 1 and 2. So type 1 will be known as von Recklinghausen. So the typical features will be neurofibromas, caveolate and leash nodules. Okay, so leash nodules are something that we can see in this diagram, we can make out from this diagram only. Neurofibromas will be, as you can see in this diagram, in the eye there will be fibroma, means growth, and there will be skin patches which is known as caveolate. Okay, that is a French term for this skin lotions or skin patches, as you can see. Okay, so these are the typical clinical features of type 1 neurofibromatosis, which is known as von Recklinghausen. Okay, and talking about type 2 neurofibromatosis, it's acoustic neurofibromatosis. So there will be bilateral acoustic neuromas. So this is basically a typical diagram given of the cross section of the spinal cord and the brain. Okay, and there will be multiple meningiomas. Okay, now what is meningiomas? Meninges, it's coming from the word meninges, which is the outer protective covering of the brain and the spinal cord. Means basically it's the protective covering of the CNS. Now, if there is any kind of abnormal growth in this meninges, then that is known as meningiomas. Okay, and multiple such abnormal growths are seen in case of type 2 neurofibromatosis, which is shown in this typical cross section of the diagrams that I have given over here. Okay, so that is the typical characteristic clinical feature of acoustic neurofibromatosis, that is type 2 neurofibromatosis. Okay, so I think that's it about the genetic basis of disease. So we talked about the different inborn errors of metabolism, and we also talked about the different sex chromosome related disorders like Turner syndrome, like Klinefelter syndrome, and we also talked about the different autosome related genetic disorders like trisomy 21, trisomy 13, trisomy 18. Okay, so I think that's it. Please do like, share and subscribe ITLS Academy. Thank you.